Okay, let's dive into this explainer and embark on a microscopic safari. Today, we're going to explore one of the most fascinating single-celled survivors on planet Earth, the Paramecium. Now, I know it might just look like a tiny speck under a lens, but as we unpack the source material today, you're going to see a highly complex, beautifully engineered organism that has completely mastered the art of survival. To keep things clear, here's our roadmap for today. First, we'll meet this slipper animalcule. Second, the anatomy of a microbe. Third, feeding and digestion. Fourth, defense and water control. And finally, cheating death, paramecium reproduction. All right, let's jump into section 1, meet the slipper animalcule. So, to start with the basics, the Paramecium belongs to the Protista kingdom, specifically falling under the ciliate class. It was actually first discovered by an early microscopist named John Hill. If you wanted to find one of these out in the wild yourself, you just need to look in some fresh, stagnant water. And if you're a scientist trying to grow them in a lab, you'd use a wonderfully traditional technique called the hay infusion method. It basically provides the absolute perfect bacterial buffet to cultivate them in massive numbers. Moving right along to section 2, anatomy of a microbe. So as you'll see, a paramecium has a highly distinct silhouette. If we compare the two ends of the cell, the anterior end, think of that as the front of the cell, is distinctly blunt and rounded, but contrast that with the posterior end at the back, which sharply tapers off. This specific asymmetry is exactly what gives it that classic slipper look that scientists are always talking about. Now, the crucial point here is how it maintains that slipper shape while zipping through the water. Its structure is held together by a thin, flexible outermost covering called a pellicle. And protruding right through this pellicle, originating from these little basal granules, are thousands of tiny hair-like structures called cilia. These cilia actually divide up the workload. You have oral cilia acting like a conveyor belt to collect food, while the body cilia act as the primary engine for locomotion. Oh, and if you look really closely at the posterior end, you'll find an enlarged cluster called the caudal tuft. This acts kind of like a set of sensory fingertips, allowing the paramecium to process touch, stick to surfaces, and rapidly change direction. Which brings us to section 3, feeding and digestion. You might be wondering how a single cell eats without a mouth. Well, the paramecium's filter feeding pathway is honestly incredible to watch. It doesn't hunt like a shark. It vacuums. Water currents are generated by specialized, highly coordinated cilia bands on the cell's surface, specifically the quadrilis and panniculus bands. These currents forcefully sweep tiny food particles into an indentation called the oral groove. The food is then pushed down into the vestibule and the cytostome, travels through a tube-like cytopharynx, and is finally packaged neatly at the base into a microscopic stomach called a food vacuole. Once that food is packaged into the vacuole, it goes on a wild ride through a phenomenal process called cyclosis, or cell streaming. The newly formed food vacuole literally streams through the cell's cytoplasm in a highly specific figure 8 pattern. This isn't just random wandering. This constant circulation ensures that digested nutrients get evenly distributed to every single corner of the organism. During that figure 8 journey, the paramecium secretes powerful digestive enzymes to break down the meal, specifically amylase for carbs, lipase for fats, and protease for proteins. But what happens to the leftovers? Well, it's quite elegant. The undigested waste is eventually ejected out of the cell membrane through a designated pore called the cytopage, which is affectionately known in the biology world as the cell anus or the cytoprac. Of course, all of these complex functions require a biological brain to run the operation. The paramecium is what biologists call heterokaryotic, which just means it splits its labor between two completely different types of nuclei. First, you have the macronucleus. It's large, kidney-shaped, and uses a type of genetic material called trophochromatin to handle all the day-to-day metabolic activities. Basically, it runs the factory. Then you have the micronucleus. It's much smaller, oval-shaped, and relies on idiochromatin, genetic material that is entirely dedicated to the task of reproduction. Interestingly enough, the exact setup of these nuclei isn't universal across all paramecia. It actually changes depending on the specific species. For example, Paramecium caudatum keeps things simple with exactly one macronucleus and one micronucleus. But Paramecium aurelia, it operates with one macronucleus and two micronuclei. It's just a fantastic example of structural diversity right down at the microscopic level. All right, section 4, defense and water control. Because these guys live in fresh water, Water is continuously rushing into the cell via osmosis. To manage that relentless pressure, the paramecium relies on a process called osmoregulation. It uses exactly two contractile vacuoles, one stationed up at the anterior end and one down at the posterior. Think of these as microscopic bilge pumps on a sinking ship. They feature 6 to 10 radiating and accessory canals that actively collect the excess water and pump it straight out of the cell, keeping the paramecium from bursting like a water balloon.
So it won't explode, which is great. But what about the threats lurking outside? How on earth does a squishy, single-celled organism defend itself in a droplet of pond water filled with predators?
Turns out they are heavily armed. Situated just below the pellicle are tiny boat-shaped structures called trichocysts. The paramecium can rapidly discharge these filaments for defense, for offensive maneuvers, and even to narcotize or paralyze their prey. But wait, there's more. Some paramecia carry a fascinating secret weapon called kappa particles. These particles consist of extranuclear cytoplasmic DNA that actually produces a lethal toxin known as paramecin. So yeah, they may be small, but they pack a serious punch. And finally, Section 5, Cheating Death, Paramecium Reproduction.
When everyday conditions are favorable, meaning optimum temperature and a solid food supply, they reproduce through an asexual process called transverse binary fission. The cell literally just splits itself in half, into an anterior half called a proter and a posterior half called an apostee. And remarkably, it takes a mere 30 to 120 minutes for this entire replication cycle to complete. So when times are good, their population can explode incredibly fast.
But, when things get tough, like when temperatures wildly fluctuate or food gets really scarce, they switch to a sexual reproductive process called conjugation. This is where two paramecia of different mating types form a temporary union. It's a pretty intense process. The everyday macronucleus completely degenerates and disappears. The micronucleus steps up, dividing meiotically to create male and female pronuclei. The two paramecia then actually exchange their motile male pronuclei, fusing them to form a brand new genetically distinct zygote nucleus known as a synkaryon. This massive genetic variation is exactly what helps the species survive the toughest conditions. And the process doesn't even stop there. After the conjugating pair separates, they're referred to as X-conjugants. That new zygote nucleus inside them undergoes a highly complex sequence of 3 rapid divisions, followed by a final cellular binary fission. The end result? Exactly 8 newly energized daughter paramecia are produced from just one single pair of X-conjugants. Because nature always has a backup plan, to survive extreme stress, paramecia have a whole suite of what biologists call aberrant reproductive behaviors. There's autogamy, which is essentially self-fertilization. There's cytogamy, where cells unite but skip the pronuclei fusion altogether. There's endomixis, a form of internal parthenogenesis. And amazingly, there is hemiexis. Now, hemiexis isn't even true reproduction. It's a purification act where the macronucleus literally disintegrates and reorganizes itself entirely just to refresh the cell's vigor without actually dividing. All of this incredible biology leads us directly to the ultimate takeaway from our source materials, summarized perfectly in this quote. Due to conjugation, there is no natural death, so paramecium never gets old. By constantly exchanging, mixing, and regenerating their genetic material through conjugation and these other aberrant methods, they continuously regain lost vigor. They are effectively hitting the reset button on their biological clocks, dodging natural aging entirely. The paramecium literally never gets old. It just keeps dividing, conjugating, and resetting itself. It's an absolute masterclass in adaptation. Which leaves us with a pretty thrilling final thought to wrap up this explainer. If a microscopic speck swimming around in puddle water can figure out the mechanics of cellular immortality, what other incredible world-changing secrets of survival are just waiting to be discovered right now in the microscopic world?